Hi everyone, and welcome back to the Chapter 8 video lectures. In the previous video, we talked about how a forensic entomologist helps identify the post-mortem interval. In this video lecture, I want to focus on that second question, determining the cause and the manner of death. Forensic entomologists can help determine cause and manner of death by using the colonization patterns of insects. Insects are drawn to fresh blood. So if there are open wounds on the body, even microscopic tears, the adult females will be drawn to those areas to lay their eggs. This gives the larva the most direct access to the nutrient supply needed for growth and development. The photograph on this slide shows fly larvae that have developed inside a head wound on the skull. If there are no open wounds, flies will lay their eggs in our natural body orifices, meaning our mouth, our nose, our ears, and our genitals. Again, these areas provide the easiest path to nutrient access for the fly larva, so they're the preferred colonization site. So let's say you come across a dead body in the woods. The body is in the early stages of decomposition and it doesn't show a lot of insect activity, but there are pockets of fly larvae in the mouth, the nose, and the chest. The mouth and the nose, those are normal areas of colonization, but it's unlikely that the flies would have colonized the chest unless there was an open wound there. This gives forensic entomologists a clue that the person probably experienced a chest wound prior to death. Now, just like post-mortem interval estimation, the insect life cycle can be used to help identify if there are wounds on the body, even in heavily decomposed bodies. Older larvae in non-orifices can indicate wounds to the skin that were used as a nesting area for the flies earlier in the decomposition process. Now, another new area of forensic entomology that is useful for cause and manner of death determinations is called entomotoxicology. This is the study of insects in order to determine drugs and poisons that were contained in the insect's food source. Say, for example, I poisoned my husband with cyanide, and then I dumped his body in the woods. When the fly larvae started eating his corpse, they will ingest some of that cyanide. If we can detect the cyanide in the fly's body, then the investigators can gain important information about the cause and manner of death that they may or may not be able to identify from the body itself. Now, I mentioned before that insect life cycles are heavily dependent on a variety of factors, and one of those is drugs in the body. Research has shown us that the presence of drugs can change the life cycle speed of insects. Some drugs accelerate the process, others drugs will slow down the process, and still others have no influence on the development rates of different larvae. Some drugs will also affect the life cycle process differently based on concentration, like methamphetamine, and that further complicates the PMI calculations. This process is not unique to fly larvae either. Drug ingestion will also affect the life cycles of adult beetles, wasps, and other necrophagous species. So for example, let's consider the impact of cocaine on insect life cycles. As the level of cocaine increases, the maggots will become more active. The larvae feed more rapidly, ingesting more of the drug, and so it perpetrates this cycle. And this behavior will increase the rate of development through the larva stages until the third larva stage. Once the maggots reach this point in their development, they stop feeding, and so the ingestion of the cocaine ceases. Instead, the cocaine begins to be metabolized by the insect, and so by the beginning of the pupal stage, the rate of development has returned to normal. In order to improve post-mortem interval calculations, researchers in entomotoxicology are working on identifying the effects of specific drugs on specific insect life cycles, specifically focusing on blowfly and flesh fly life cycles because these are the most commonly used for time of death determination. By charting out each drug's impact, they can better improve post-mortem calculations and provide investigators with more precise data. Another emerging field within forensic entomology is the use of insect larvae to collect DNA samples from corpses. The hope is that by using insect samples, investigators can identify victims who are too heavily decomposed to allow for DNA analysis, as well as victims whose bodies have been scattered or disposed of. This is an emerging field because not only is DNA a more recent analysis technique, but previously we needed large samples of DNA to obtain full biological profiles. With new technological advancements, we're able to analyze very small samples of DNA through a copying process called short tandem repeat. We'll talk about that in the next couple of video lectures. So even if the insect only contains a very small amount of decomposed material, we now have the technology to actually analyze that by copying that into a sample large enough to analyze. Research in this area has suggested that DNA degrades very quickly inside an insect body. In 2016, several researchers attempted to recover human DNA samples from maggots who were fed on decomposing bodies. They used this STR analysis to assist in the DNA recovery. The researchers had two different groups. The first group was fed on the decomposing human flesh, and then it was moved to a beef source. The second group was fed on the decomposing flesh, and then it was moved off of it, but it wasn't given another food source. The researchers found that the human DNA could be identified for up to 2 days for the maggots that moved to the beef, and up to 4 days for the maggots that were not provided with a second source of food. They also noted, though, that the DNA samples they collected were very degraded and were only allowed for the development of a partial DNA profile. The hope is that with continued research into this area and with more advances in technology, we can eventually obtain complete DNA samples or complete DNA profiles even after long periods of time from insects. All right, well, that concludes the Chapter 8 lecture videos. Thanks for watching.